I already mentioned epitope tags. So again, as mentioned, these are very small amino acid sequences that are added to your protein of interest. The nice thing here is there's less chance of getting the artifacts that you'd have from a large tag, such as a GFP molecule. They can be used in repeat sequences. So for example, there's a HA tag here. Very often on a plasmid, we'll see 3x HA, which is basically this HA sequence repeated 3 times. The benefit of this is it gives you more sites for labeling with antibodies and detecting those HA molecules. So you can see down here, our GFP molecule is 238 amino acids. That's 27 kilodaltons in size versus a MIC or a HA tag, which is probably about 10 to 12 amino acids in length, more like 1 to 1.2 kilodaltons in size. The downside with the epitope tags is they require labeling afterwards with either an antibody or a dye to actually create the fluorescence. They don't have inherent fluorescence themselves. Okay, so how would we do that in terms of antibody labeling? So there are 2 different ways that we can approach this. One is called a direct immunofluorescence assay. So again, using an antibody molecule here to detect our protein of interest on a sample. So these little green things here are our protein of interest, which are being detected by the variable domains on the antibody. In this case, this is conjugated directly with a fluorophore. So this is what we call the direct labeling method. If we use the second approach, which is an indirect method, then again we have our primary, what we call our primary antibody, which is binding to the protein of interest. In this case, it doesn't have a fluorophore attached. What we're doing is we're coming in with a secondary antibody, which is set up to detect the primary antibody, and it is the secondary antibody that has the fluorophore attached onto it. So the nice thing here, although it's not shown in this diagram, is that we can get multiple secondary antibodies to bind to a primary, and therefore we're amplifying up the signal. Okay, in terms of primary labeling, so there are different types of antibodies that are available. So polyclonal or monoclonal. So monoclonal suggests that that antibody is only detecting to one epitope within your protein, so one binding position. If you have a polyclonal antibody, then the antibodies within that mix will bind to multiple locations across your protein of interest. And again, monoclonal can be very clean, but won't necessarily give you very large signal. Polyclonal can be a little bit dirtier, but will give you much larger signal amplification for your detecting your protein of interest. We need to be aware of the isotype of the antibody that you're using. We also need to be aware of what animal it was raised in, especially if we're using the indirect method, because we need to know that for our secondary antibody that we'll be using. One other thing that's coming more and more into use now is the use of nanobodies. These are very, very small antibodies from animals such as camels, alpacas, sharks, that type of thing. And these are very useful, especially when we come to do things like super resolution microscopy. In terms of where you would get those from, a lot of the common targets are available commercially through companies such as Abcam, or you can send off to have antibodies raised commercially, specifically against your protein of interest. Back when I was a student, you used to home make your own antibodies, but I'm not sure that that's really something that happens anymore in universities. So mostly it's through the commercial routes. I mentioned secondary labeling, or the fluorophore that's attached to your antibodies that you're interested in. So again, like the fluorescent proteins, we now have a vast array of different fluorophores which we can attach to these antibodies. And again, that allows us to do multi-channel imaging with different controls and things within your experiment. So we're very fortunate now to have a full range running from the ultraviolet into the near infrared, and it's going even further into the infrared now. We mentioned photostability earlier on in terms of the fluorescent properties. So this is just an example of this. So this is time over here and fluorescence. So just to show you that if you have fluorescein sitting in a drawer somewhere, it's probably not the best fluorophore to use in terms of its stability over time. Whereas something more like an Alexa fluor dye has a lot more stability over some of the older first established dyes. So yeah, just be careful in terms of what fluorophore you're selecting for your experiments.